Hi, so my grade 10 lesson that I chose to present today is the one on cells. And cells falls within the life at a molecular, cellular and tissue level and is on page 25 of the CAPS document. It's going to be a 60-minute lesson, and at the school that I teach, on average we have about 24 learners in a class. So for the purposes of this life science lesson, there's going to be 24 learners. So if we're talking about cells, learners have some prior knowledge. In grade 8, they were taught a little bit about microorganisms and that they're usually unicellular organisms. And then grade 9, they go deeper into the cell, which they started distinguishing characteristics between plant and animal cells and the basic organelles such as a nucleus, that there is a cell membrane, a cell wall, that type of thing. And while this lesson covers cells, it's impossible to cover all of cells in the grade 10 syllabus in 60 minutes. So my lesson focuses on one component, which is the cell membrane and the fluid mosaic model. So this would be the second lesson in the series of cells. So in the previous lessons, the grade 10s would have covered the cell wall, what is the structure and the function, and they also would have started the lecture, or the lesson rather, with microscopy, which microscopes are needed for what, etc., etc. So there are several learning objectives. The knowledge objectives is they have to be able to define a semi-permeable membrane, which is a very important term because from grade 10 all the way through to the end of their matric syllabus, they need to understand a semi-permeable or selectively permeable membrane, as the term comes up often. They also have to understand the fluid mosaic model, how it helps the membrane to do its job within a cell, and describe the three modes of transport that enable molecules to cross the membrane. They're going to be performing two skills in this lesson. They have to draw a scientific diagram as well as construct a model, and then they should, at the end of this lesson, be walking away with appreciation for the complexity of something that is microscopic. It's so small, and yet it is so intricate to be able to perform its function. So it really, I'm hoping there's a wow factor that the learners would walk away from this lesson. So when we start this lesson, I'm not going to go through the assessments and the resources because that they are typed out in the lesson plan. So I'm going to get straight to the lesson. So what the learners would have received in their previous lesson is a printout of all the slides. So the slides for all the cell components, and they would have covered the cell wall in the previous slide. So starting with this lesson, they would be using the slides particularly pertaining to the cell membrane. So as the introduction to the lesson, it's roughly about 5 minutes. The teacher would greet and seat the learners, and once everything is quieted down, she basically, or I, in this case, I would recap the previous lesson by asking learners questions that are supposed to encourage engagement. So things like a comprehension type question is, what type of microscope would you use to view a virus? So learners must think that a virus is too small to be able to be seen with a standard light microscope, so you would need an electron microscope. Straightforward knowledge questions like differences between plant and animal cells, what is the function of the cell walls, and then possibly even an application type question, why do animals not have cell walls? Animals typically have a skeleton, hydrostatic skeleton, exoskeleton, endoskeleton, so they don't need to rely on cell walls to be able to give them structure. So the learners would have to think about that reason a bit for why animals don't have cell walls. After we've recapped the previous lesson, we can start the actual lesson for today, which is the cell membrane. It's roughly about 50 minutes, and I would ask the learners to please turn to the correct page in their printout of the slides, and I would portray the slides on the overhead projector on the whiteboard. So for the first few slides, which are also one of the appendices at the back of the lesson plan, I would first talk about the cell membrane, other terms that you could use for it so they could recognize it in textbooks or if it comes up in the question. The fluid mosaic model, which is the term, or the theory rather, that we use to study the cell membrane. And then the fact that it's the outer layer of the cell. And at this point I would like to pause because this is where we get into the definition of the semi-permeable membrane, which is crucially important that the learners understand. Excuse me. So learners would have to understand that there are certain molecules that can freely cross the membrane. There are some molecules that need assistance and energy to be able to cross the membrane, and other molecules are denied access to the cell, so they can't enter at all, which is essentially what defines a semi-permeable membrane. So at this point I would discuss it a little bit with the learners. We'd give possible examples of molecules that can either enter freely or need transport or aren't able to enter into the cell. And once I'm happy that the learners understand the term selectively permeable, I carry on from slide 5 all the way to slide 12. Now those slides detail the structure of the fluid mosaic model. The terminology is there, the descriptions are written out in words, and there's lots of images, different images of the cell membrane, so it can appeal to those learners that prefer reading as well as those that are more visual and prefer images. And then obviously we would discuss it, so we would say there's a bilayer of phospholipids with hydrophobic tails, hydrophilic heads. Embedded in this bilayer there are proteins which are carrier or transport proteins, and they help molecules to cross the membrane if they are not able to do so freely. Different ways are applicable. There might be a channel through the protein, or something might bind to a protein and the protein flips until it's on the inside of the cell. So we can discuss those. And then the glycolipids and the glycoproteins, which are attached to the carbohydrates in the membrane, those are there for cell integrity, to maintain the membrane integrity, so the membrane doesn't break apart. It's also for cell recognition, so that your immune system does not destroy your own body cells, but recognizes it as your human cells, as opposed to, for example, a pathogen. Once we've gone through the structure, the learners need to be able to draw the diagram. So at this point, I would draw a diagram of the cell membrane, the fluid mosaic model, on the whiteboard, and then I would refer learners to their textbooks. On page 58, there's a diagram as well. So the learners are welcome to use my diagram on the board or the one in their textbooks. It has to be drawn in the workbook. This is individual work because they all need to be able to draw the diagram, and it has to be drawn scientifically correct, because in life science all diagrams are correct. So I will remind the learners that they need a heading. It needs to be drawn in pencil, and it needs to be drawn with clean lines, so there's no sketching or shading, that type of thing. Label lines and labels must be in pen, no arrowhead. The labels shouldn't cross one another, they must be parallel, labels neatly beneath one another. And while the learners are busy with this, I would walk around, make sure that their drawings are correct, that their skills are correct, that type of thing. After that, once the diagrams are complete, if there are learners that are taking a little bit longer than normal, they can complete their diagrams for homework so that we don't fall behind schedule. And then we will carry on with slide 13, which discusses the transport across the membrane. Now there are 3 main types of transport: diffusion, osmosis, and active transport, and they need to understand all of those. So for diffusion and osmosis, diffusion is the movement of a liquid and a gas from a high concentration to a low concentration, down a concentration gradient. And osmosis is the movement of water from a high concentration to a low concentration through a semi-permeable membrane. So there are nice demonstrations to demonstrate these processes. So for example, for diffusion, you could spray deodorant can at the beginning of the lesson, at the front of the class, and you could note when learners are able to smell it, so clearly it's moving back through the class. Or you could put a drop of food coloring in a beak of water and watch it disperse. For osmosis, you could put a potato or an egg in hypertonic, and isotonic solutions and watch as it grows or shrinks. But there's not enough time in this lesson for those demonstrations. So either they would have to be conducted in the following lesson, or perhaps in future lessons, the diagram that they'd previously drawn could be assigned for homework, and that would create a bit of space within the lesson to do these demonstrations as well. Then active transport, where molecules are moving against a concentration gradient, and they need protein molecules or carrier molecules to help them cross the membrane, and typically they also need energy for this process. At this point, learners would be divided into groups of 4, where they would be constructing their model of the fluid mosaic model. And constructing a model is very hands-on. It benefits both the kinesthetic and the visual learners. They can see how the different components work together and fit together. And working within groups, they're able to explain the terms to one another, possibly in words that the teacher didn't use, which might be easier to understand for those that are struggling to follow in the lesson at this point. And also the groups are small enough to make sure everybody is engaged and included within this activity. So they need bar ones, sour worms, jelly tots, and marshmallows. constructed a model over here. Let me get a little closer for that, of what their fluid mosaic model should be looking like after they've constructed it. And after the learners have constructed it, the teacher or myself will walk around and ask them to explain what the different functions are, how they work together, how do the three modes of transport enable molecules to cross the model they've constructed, and that type of thing. So hopefully at the end of that model construction, they really do understand the fluid mosaic model. And then for consolidation, which is the last 5 minutes, similar to the introduction, I would just briefly recap, mainly by asking learners questions, so it remains interactive and they remain engaged in this lesson. So questions like, what is the function of cell membrane, define osmosis, so questions that they haven't just explained to me in the model construction, but other questions pertaining to the lesson. And then there would be no homework assigned for this particular lesson. So there are many questions going to be asked in this particular lesson. They engage on all the taxonomies of, all levels of Bloom taxonomy. And for learners that require special attention, it's not really necessary in this specific lesson, because even though there are learners that aren't dyslexic, there's no need to write many notes in this lesson. It's more a hands-on activity, so it's not going to disadvantage them in any way. So the advantages of this lesson is obviously that it's fun, learners enjoy practical hands-on activities, it engages all the different types of learning, visual, oral, reading and writing, kinesthetic, all of that is available for the learners, so whichever way they process information best, hopefully they are able to understand and access the information. Also, they get to eat the sweets. Learners always enjoy eating sweets, and there's group and individual work. And then the disadvantages. In South Africa, we always have to take into mind load shedding, but the only part in this lesson which uses electricity is the projector, and the learners have the printouts of the notes. So if there is no electricity, it's fine. The lesson can still continue without any hassle. They could be very noisy. Group work always ends up noisy, which means you need strict discipline. But that would have to be in place from the beginning of the year already. You can't only implement it within this one lesson, or the learners wouldn't respond to you. So discipline needs to be maintained throughout the year so that you are able to conduct lessons such as this one. And also then the matter of the sweets. If learners had to bring the sweets themselves, you also have to prepare for those learners that forgot or were unable to bring sweets. So have a couple of backup, or if you provide the sweets, make sure you have enough sweets for everybody to be able to construct their model. I hope you enjoyed the lesson. Thank you.